you everybody for coming and to what is the last online and cell size and growth seminar for this year. We're very lucky to have two speakers who are going to talk to us about different aspects of growth regulation and how protein production is connected to aspects of cell physiology. We're going to start with Hisao from Okayama University, who, first of all, I'm very grateful for coming to speak to us at what I think is about midnight his time. So without any further ado, I'm going to stop sharing this and I'll ask you to share your slides and get us started.
Okay, I'll share a slide and I hope you can see it, right? So thank you very much for inviting me this great meeting. Today I'm going to talk about the impact of maximal overexpression of a non-toxic protein on yeast cell physiology, which is recently published in eLife. And I'm Hisao Moriya from Okayama University in Japan. And first part, I would like to introduce what is a non-toxic protein, and then I will introduce what could happen when it's ultimately overexpressed. So this is a basic introduction. So showing the protein level of yeast cell. And this axis shows the ranking, and this axis shows the protein level. And as you can see here, for example, glycolytic proteins and ribosome proteins are really highly expressed, about 100,000 copies, more than 100 copies per cell. And some, on the other hand, cell cycle and transcription factors proteins are not so much expressed, like 10 to 100 copies per cell. But you can see the protein expression levels tend to correlate with their functional categories. So which means it's not randomly distributed. It should be there should be some evolutionary bias to determine the expression level. Then what determine the expression levels of these proteins? And there's one interesting experiment performed by Karen et al. From Weizmann Institute in 2016. They changed the promoter of yeast genes using synthetic promoters with expression spans 500-fold range, and then integrated into the upstream of the target gene, and measure the effect of the changing in gene expression. So this is a typical example. For example, you get this red line shows the normal expression level, and if you decrease or increase the expression level, you observe the reduction of fitness. So by this kind of experiment, you can obtain the expression fitness relationship. And probably you can imagine there should be this kind of relationship because native expression levels should be optimal to perform the highest fitness. And if you reduce the expression level by knockdown experiment, or if you overexpress the expression level by expression experiment, you observe the reduction in fitness. And if you observe this kind of fitness relationship, then you can imagine there should be an evolutionary bias. The one evolutionary bias is demand, which should increase the expression level because of the functionality requirement of the protein. And if you overexpress the protein and observe the reduction in fitness, you can imagine there should be some evolutionary bias collectively called constraint or cost or burden, and which is triggered by the toxicity of the protein. And if you observe the reduction of the fitness, the expression level that causes growth inhibition is called expression limit in this talk. And the reason why expression limit or toxicity is triggered by the overexpression can be explained by the fate of protein. So protein should be transcribed and translated, folded and degraded and localized, and it has activity and it has interaction partners, and the overexpression causes the trouble in these processes. And the trouble or constraint can be divided into 4 different mechanisms: resource overload, stoichiometry imbalance, pathway modulation, and promiscuous interactions. I don't have enough time to explain in detail of all 4 mechanisms, so I just want to focus on one constraint, resource overload. which is triggered by the overloading the capacity of processes, protein process, protein, for example, synthesis or localization, degradation, etc. So if you overexpress one protein, it should monopolize and deplete and compete for resources. And the resource overload can be understood by another model. That is so-called, I call it a barrel model for resource overload, in which the size of barrel indicates the capacity of process, and the arrows indicate the proteins to be processed. So we can imagine the synthesis process. Virtually all proteins are processed through this process. So the synthesis process should have the biggest capacity, and transport capacity and folding and degradation capacity should be smaller. And if you, for example, overexpress this protein, the first process overloaded should be transport rather than synthesis. And this can be considered as a bottleneck to be overloaded by the overexpression of red protein. This kind of concept is actually tested by an experiment. This is our previous paper. And we overexpress, for example, GFP or MOX GFP. They are basically localized to the cytoplasm. And you will observe the expression limit is about 15% of total protein. But if you add the localization signal, like mitochondrial localization signal, nuclear export signal, or vesicle-mediated transport signal, then you will observe the reduction of the expression limit because the transport capacity is smaller than the synthesis capacity. And if you expand this idea, you can imagine if one protein doesn't localize, doesn't have activity, and doesn't have an interaction partner, and it's not degraded actively, and it folded itself, that this part is remained, and only actually this protein can overload the synthesis process. And the overloading of the synthesis process is especially called like a protein burden, and that's triggered by the monopolization, depletion, or competition for biosynthetic resources. And previously, we've found that we believe that the 15% of total protein, if you overexpress one protein, then you can overload the protein synthesis process. And let me go back to the first model. So the protein expression level is determined by constraint, demand and constraint. But constraint is actually not a single layer. There should be a hierarchical layer exist, and then if you remove one by one, so you observe the increase in the expression limit. For example, activity is removed, localization is removed, then finally you reach to the expression limit by overloading the synthesis process, which is I call here protein burden limit. So this kind of protein can be considered as non-toxic protein, as you can express really high level and doesn't cause any trouble other than synthesis process. Then what are proteins, non-toxic proteins? So previously, actually 3 research groups, including our groups, analyzed the protein burden using fluorescent protein. Like Nama-Barker group used M-Cherry, and the Farkas and Papp group used Venus, and my group used actually EGP, by believing that they are non-functional in yeast cell, and there's no interaction partners, and not localized, and fold themselves, and not actively degraded. So we believe they are good model proteins for protein burden research. However, actually we found that the overexpression of Venus or EGFP causes the abnormal cell elongation and overloading of chaperone system. So we believe that they are non-toxic protein, but they actually perturb the folding process. So they are under the folding constraint. and not good model protein for the protein burden research. So we changed the target protein to MOX-GFP, which is basically the superfold derivative without cysteine, and it overexpression didn't cause any wrong morphology and no detectable burden for chaperone. However, we accidentally actually found when you remove the fluorophore from by mutating the fluorophore amino acid, and the fluorescence loss actually increased the expression limit up to threefold. And so namely, actually a fluorescent protein itself has some sort of toxicity by fluorescent, and the expression limit can be increased after removing the it's a fluorescent activity. So we now we believe that we reached to the synthesis protein burden limit, and then what did we learn from the finding? So now on, I will go into the detail of experimental data. Okay. So by overexpressing the non-toxic protein to the protein burden limit, I actually need a really strong expression system, which we call genetic tug of war. And in genetic tug of war, we use one plasmid, and in this case, we used really strong promoter, strongest promoter in each cell, and we use this plasmid. And this plasmid is actually multi-copy plasmid, and there is a neutralient bias to increase the plasmid copy number up to more than 100 copies. So basically, inducing minus condition, this plasmid copy number becomes more than 100, and you express the target protein using TDH3 promoter, so it expresses really strong level. And this is initial finding. This is actually my notebook, and I changed the tyrosine in the fluorophore of MOX-GFP to glycine to remove the fluorescent activity. Then the expression limit becomes actually threefold higher than the normal one, the fluorescent one. So we started the systematical analysis of some fluorescent proteins like EGFP, superfolder GFP, MOX-GFP, and MHD, and mutated the fluorophore of those proteins. And in every case, in all cases, the expression limit is actually increased like this. However, the growth rate are somewhat different. So we decided to calculate and make a index indicating the toxicity or neutrality of the protein. So this is actually the calculation. So we use the growth rate and the protein level, and multiplying the growth rate and the protein level, we calculate the area of this area, and then this area indicates if the area is wider, the protein should be more neutral, which means non-toxic, and this area is smaller, then the protein should be more toxic. And by this calculation, we got a neutrality index, and MOX-YG, non-fluorescent MOX-GFP, is the most neutral protein. And this and through the analysis, we found one important finding that previously, actually, overexpression of MHD causes a cell size increase. So that is believed that this is a response on the protein production, perturbation on protein production, because the cell requires to synthesize more ribosome or other synthesis machinery, then the cell size would become larger. But it was actually only phenotype found in the fluorescent MHD. If you mutate the fluorophore, then you do not observe the cellular enlargement anymore. So cell enlargement is not a common phenotype under protein burden. And we've studied several omics analysis. This one is a transcriptomic analysis of the cells of expressing MOX-GFP and MOX-YG. And we found that the overexpression of MOX-YG, for example, overexpression of MOX-GFP, we observed the increase in the ribosome biogenesis. However, if you overexpress MOX-YG up to 43%, then you do not observe the ribosome biogenesis increase. Actually, the reduction of ribosome biogenesis is observed. And we've also observed the induction of some amino acid transporter or ammonium transporter upon overexpression of MOX-YG, so indicating we finally observe the depletion or competition of synthetic, namely translation resources. And also we performed proteome analysis, and importantly, the total protein level in the cell are consistent. If you overexpress MOX-YG, then the protein, normal proteome, actually proteome size becomes half of the normal proteome, like this. So the proteome is shrinked to the half of the normal proteome, and the contents of some categories are different, changed. For example, the expression of mitochondrial proteins are dramatically increased, and the ribosome biogenesis and the ribosomal proteins and glycolytic proteins are collectively reduced. So it seems like a metabolic shift from fermentation to respiration, and a decrease in ribosome abundance are observed. And those proteome change are quite similar to the response upon TORC1 pathway inactivation. So probably massive protein overexpression will cause the anosose depletion, and it causes the inactivation of TORC1 pathway, and maybe cell goes into the economy mode. And we also found several interesting phenotypes. The one of it is this one is electron microscopic images, and we observed that the dilation in the cytoplasm, these black dots are ribosomes, so cytoplasmic ribosome density are reduced, which is predicted by from proteome analysis. And also we've observed the nucleolus formation. Here the nucleus and nucleus. Nucleolus formation is somewhat wrong. So the nucleolus are dispersed in the cell, and as nucleolus are created by the lipid phase separation, so the dilation in the cytoplasm or nuclear protein contents will cause the disruption of the nucleolus. And we have also observed some reduction in the top speed growth or the high temperature sensitivity upon overexpression MOX-YG. Those are not explained yet, but we have to analyze more. And finally, there is actually a remaining question that why does fluorescent limit expression? And we don't have any idea yet. We cannot concrete evidence yet. So it's not due to the phototoxicity or loss of tyrosine. And loss may be involved, but there's no evidence for the oxidative stress response. So I doubt it. And the maybe protonation of the active center might be involved, but we still don't know why the fluorescence could cause the toxicity. So finally, I would like to thank my laboratory member, especially Yuri Fujita, who performed most of our experiment presented here, and these grant agencies. So thank you very much for listening.
Fantastic. Thank you very much for it was a really interesting talk. I'd like to just remind everybody that if you have questions, in the next 10-15 minutes, we'll do a Q&A. If you have questions, please place them in the chat, and I'll call on you to ask your question. I'm gonna start and ask you to just elaborate a little bit more on the comparison you made with this work from the Buckeye Lab, where one thing that they did is in different nutrient conditions, they assigned the burden in their context to transcription or translation.
Yeah, yeah,
Obviously,
yeah.
you have this significantly improved system where you're really focusing just on the synthesis. If you do
Mm.
that same type of analysis, what kind of conclusion do you come to with your system?
Actually, we didn't, namely, you are mentioning the phosphate limitation and the nitrogen limitation experiment, but we didn't do that. But we found that uh, we did which synthesis, transcription or translation are perturbed in this experiment. To test it, we made frame sheet mutants. The frame sheet mutant actually can transcribe, but it doesn't cause the trouble in translation. And that mutant completely uh, recovered. Namely, the transcription is not so much, it has much bigger capacity, we believe, and the translation is part of the in our system.
Yeah, that makes
Okay.
sense. And so I
Mm.
think Hui has what might be a related question, but Hui, I don't know if you'd like to unmute and elaborate a little bit.
Hi, thanks for the talk. So I was just wondering because the fraction of ribosomes and translation capacity varies
Mm.
between um, nutrient conditions and growth
Mhm.
rates in general. Do you think, or are you interested in doing an experiment where expression limit might change? Because, for example, there are fewer ribosomes, so cells are more
Mm.
sensitive to toxic proteins that it normally would do better. Thank you.
So you mean higher nutrient condition, the effect of higher nutrient condition, right?
Yeah, for example, for the same protein,
Mm.
is the expression limit lower if we grow it on ethanol and glycerol instead of glucose,
Yeah,
something like that?
we haven't tested the different carbon sources, and however, looks like as I showed almost last slide, but the early growth phase, like glucose growing cell, seems to have a trouble upon overexpression of MOXYG. But later, the after glucose is almost consumed, the growth rate is quite similar, which means the the cell is really highly growing early phase of the log phase. So in that case, it seems to be trouble. And then slowing down the growth rate, then it looks like okay. But and what we are actually want to do is to use the much more richer conditions like YPD, so-called rich medium, and we are now developing the expression system to test in the rich medium. So I hope I can get the answer for you.
Thank you.
Great. Next up, we have a question from Nick.
Yes, I was just wondering to what extent is simply the physical size and volume of the overexpressed protein a factor here? The dilution of ribosomes being just because there's no room for them because all of that space in between them is taken up by your non-toxic protein.
Yep. Well, that's very nice, very good question. And you mean the cell has a volume and the protein should be packed in the cell, and then other protein should be reduced. And importantly, what we think is the difference in the MOX, fluorescent MOX and non-fluorescent MOX might be the rigidity or strength of the structure. So the fluorescent protein might be much more harder or rigidly folded because it has the tyrosine inside. And then if you remove it, the protein should be softer than the fluorescent protein. Then the protein can be expressed more because it doesn't so much perturb the cytoplasmic, I mean, the molecular crowding. So then that protein can be expressed threefold or more or something. So that's not we are focusing on because it will cause the molecular crowding and the difference between the MOX and the MOX-YG. Is it
Okay.
okay? Is that answering?
Yeah, thank
Yeah.
you.
Okay.
Great. Next up, we have a question from Bruce.
Hi, can you give us some more details about how the proteins in the proteome were measured for the overexpressing strains? And I'm particularly interested in whether the results are absolute numbers of molecules of protein or
Mm.
or some kind of relative measurement relative to something.
It's relative measurement, and the normalization is sort of important. And in shown data was actually normalized. The fraction, it's basically the proteome size is like a shrink to the half. So we increased to compare the contents between vector control and MOX-YG. So we increased twofold in total and then compared. In that case, the ribosome contents are still reduced and mitochondrial contents are increased. Okay.
Okay, and what is the method of measurement? Is it mass spec or
Ah,
something else?
it's mass spec,
It's mass spec.
and DIA proteome analysis.
Okay, thank you.
All right. Jan's up next.
Yeah, so I was wondering also if you could tell us a little bit more about the mass spec results. And so I was assuming that as you generate this protein burden cell, that you elicit the general stress response.
Yeah, yeah.
So, and this is like
Mm.
nothing particular
Mhm.
about the proteome that you see. It's
Yeah.
just the normal thing that happens as you slow
Mm.
down
Mm.
growth.
Yeah, that's what we actually observed, and it's the reason. Maybe the general stress response is somewhat quite overlapped with the inactivation of the TOC1 pathway. So that's why we are saying that actually I'm saying that it's the TOC1 the inactivation triggers the stress response. Okay.
Okay.
So I had a kind of a connected question, which actually goes back to how my first question started, which was again this comparison with the Baikal lab. They did an analysis like that in their conditions, where they threw out this correlation with mediator phenotypes. They said that there was this mediator-like phenotype in all of their burden cells, and there were flocculation defects, and
Mhm.
the transcriptional signature was more associated with a mediator deletion than it was with a general.
Yeah.
So,
Yes.
would I be correct to assume that you don't see any of that in your system as well?
And we didn't observe that kind of, namely transcription perturbation, we didn't observe. So maybe, I don't know what is the difference but at least in our system, so transcription it seems to be totally okay. And instead of translation. So,
Yeah, super interesting. Okay, so next
mm,
up we have a question from Drish.
Hi. You showed your previous results with like mitochondrially targeted proteins that showed that if you overload the kind of transport barrel, that the protein amount goes down. But this doesn't seem immediately obvious to me. So do you generally see this kind of, feedback from later barrels in your model to synthesis or is it to degradation? Like when you have this kind of reduction in protein amount, is it because they're being synthesized less or they're being degraded more, or both?
what, what, I cannot catch your question. It's not a reduction in
Yeah.
the protein. It's actually the limit of protein expression. So it will cause growth defect so much. So that's why the protein expression limit should be lower. Because if you try to increase more, then the uh, cell will be dead.
Okay.
Okay. So there's no feedback system. It's just it's observing the limit. It's not the expression level
Okay.
itself.
Mhm.
Okay, well, fantastic. I think that concludes all of the questions we have for this first half of the seminar. I'd like to thank Hisao again for a really wonderful and interesting talk. And then we're going to continue with Jin, who's a postdoc in Jan Schottheim's lab at Stanford, who's going to talk to us about the scaling of RNA and ribosome concentrations during cell growth. So, Jin, over to you.
Can you see it?
Yep, looks good.
Yeah. Okay. Hi. Wait. Okay. Hi everyone. Today I'm going to show you something about cell growth, how ribosome and mRNA concentration set the growth rate. We know we know cell growth is very important because it's cell need to growth and to some size and triggers division. So it's almost the fundamental of all aspects of cell physiology. So in the like 15 years ago, Terry Hua they found that ribosome concentration is linearly correlation increase with growth rate, and they call it like bacterial growth law. And they build a very elegant model. They build some location model. For example, this part of protein goes to ribosome. This part of protein goes to like uh, metabolism and and there's a fraction of another model. So they actually find how the bacteria cell allocates ribosome source to set the growth rate, but this model actually have nothing specific to yeast. So we want to ask whether this is also work in budding yeast, in eukaryotic budding yeast. But we face some difficult when we transfer the model to the budding yeast. First is there are a lot of like inactive ribosome in budding yeast. Like 10 years ago, Nama Bakai lab, they use polysome profile to study how the active ribosome fraction change. They just assume all the polysomes are active and all the monosomes are inactive. So you can use the fraction of polysome to all the ribosomes to as a proxy of active ribosome fraction. But they only did this in two conditions, and some papers show that monosomes can also be active, especially in those slow growth condition, right? So we probably need a more accurate method to determine the active ribosome fraction in eukaryotic yeast. And most of studies they just assume all ribosomes are active. So actually, I don't think it's good enough. And other than active ribosome fraction, we also have no idea of peptide elongation rate of a single ribosome because almost 50 years ago, like two paper used radioactive post-chain labeling, and some paper found it's constant, some paper found it's like increase with growth and only have like few data point. So I think so to understand how yeast cell set their growth rate, we have to know the ribosome concentration, how the ribosome concentration change with growth rate, and also study how the active ribosome fraction change with growth rate, and study find how peptide elongation rate change. So we start with measuring these very fundamental parameters first. Here, we use label-free mass spec and to mention the ribosome fraction of proteome in 15 different conditions. And if we plot them with growth rate, you find it's linearly correlated with growth rate and very close to the to other paper. And RNA to protein can also be a proxy of ribosome concentration because most of RNA are RNA. So like we mentioned the mass of RNA and protein, and you can find they have almost the same trend with the data from mass spec. So we think that ribosome is increased with growth rate, which is same with E. coli. And next step, we have to study how the active ribosome change with growth rate. So here we use single molecule tracking. We fuse the halo tag to the 48 subunit. When the ribosome is inactive, it's in the cytoplasm and it's diffused very fast. When once it's active, it's binding with mRNA, and mRNA is quite large compared with ribosome. So we think the diffusion coefficient will sharply decrease, and you can determine the active ribosome fraction based on the fast and the slow two groups of the ribosome, right? And now we do the single molecule tracking of the ribosome, and you can see each particle denotes a ribosome, and we will track the trajectory of single ribosome and then calculate diffusion coefficient of ribosome. And we can plot them together, and you can see in different different media, the growth rate is gradually decreasing in this direction. And you can find if we use Gaussian mixture model to fit the distribution, you can find the active fraction, the left peak denotes active. The active fraction is gradually decreased with growth rate. And we can, like showed, it put it here. We can see the active ribosome is gradually increased with growth rate, and no matter in both carbon limited and nitrogen limited conditions. And if we times the active ribosome fraction with ribosome concentration, which mentioned from mass spec, you can know the active ribosome fraction in the proteome. We find it's like just linearly correlated with growth rate, and there's like the inactive ribosome of the proteome is slightly decreased. And with this measurement, and we can start to calculate peptide elongation rate of a single ribosome is just equals to the protein mass increase in the cell in a cell cycle over the ribosome number times like active ribosome fraction, something like that. And we can find the peptide elongation rate of a single ribosome is almost constant in different growth conditions. Like this is very surprise, because in E. coli we know the peptide the elongation rate is gradually increased with growth rate, because tRNA have some limitation to the peptide elongation rate. But in our result, the peptide elongation rate is constant. Okay, and if we assume like all ribosomes are active, so we can calculate the average amino acids make per ribosome. So it's very similar with previous reported data. Yeah, so okay. So we further do some tests to our methods. So now here we add the cycloheximide, which is a translation inhibitor, and it's like blocks the elongation process, right? We use a very low concentration of cycloheximide, and we do mass spec, we do a single mode tracking to determine the active ribosome fraction, and we can calculate the peptide elongation rate with this cycloheximide. You can see that with cycloheximide, the peptide elongation rate significantly decreased. And if the higher cycloheximide concentration will lead to slower peptide elongation rate, which proves our method is solid. And so now we want to ask, now we want to ask why the active ribosome fraction changed with growth rate, why it changed, right? Because we think ribosome binding with mRNA, so we have to mention the total mRNA concentration first. So we use the spiking RNA-seq to determine the total mRNA concentration. Like you just add some Sigabrada sample to your binding sample, and you can calculate the fraction of the use Sigabrada as a benchmark, and you can calculate total mRNA concentration, and the time and use the mRNA number over cell size, and you can know how the mRNA concentration changed. And we found the mRNA concentration is increased with growth rate. People usually think the mRNA concentration is always constant. It's just scale with cell size, but we found it's increased with growth rate. But for each gene, the mRNA fraction is always like very close to the protein fraction, but the total mRNA concentration is increased with growth rate. Okay, now we spend some time to explain why total mRNA increased with growth rate, because our mass spec data shows the POR2 concentration is just a slightly increased with growth rate, and the decay rate is almost the same in different media. So this is very confused. And here we found the nuclear DNA concentration change with growth rate, and we do some single molecule tracking of RPB1, is the largest subunit of POR2. We found in fast growth condition like glucose, like almost 58% of POR2 bound with DNA, and in slow growth condition like only 30 in trehalose, only 30% of POR2 bound on DNA. So this is very interesting, because like in fast growth condition, more POR2 are active, right? So here we introduce Matthews Matthews model. It's simple. Just consider how the POR2 binding with DNA and export out of the nucleus, and we find this model can fit well with our experiments. Yeah, and it explains the port to bond fraction increase, and this lead to the total mRNA concentration increase 4 times. Okay, so now we know the mRNA concentration increase with growth rate, and we can we know mRNA concentration increase with growth rate, and we know ribosome concentration increase with growth rate. So we can try to explain why the active ribosome and the growth rate changed. Here we build a very simple mRNA and the ribosome dynamics. Just consider how the mRNA binding at the initiation site, and after sometimes it's like initiation, and the binding site is free, and it can bend new ribosome, and it has the constant rate dissociation. And after the dimension length of this model, we only have one parameters here need to be fitted, right? And we fit this parameter, fit our model with the single molecule tracking data, and we can find that the active ribosome fraction fit well with our model, and it's like with more mRNA, the active ribosome fraction will gradually increase. Yeah. And we have some tests for our model because we said mRNA concentration influence the active ribosome fraction and then change the growth rate, right? So we try to decrease the total mRNA concentration and find how it's influenced the active ribosome fraction. Here we replace the promoter of RPB1 to ATC-induced promoter, so we can give different drug concentration and regulate RPB1 expression level. And we find in different ATC concentration, the total mRNA concentration increased from 0.4 to like 1 times of the original level. And we mentioned the active ribosome concentration with single molecule tracking, and we can find the active ribosome concentration actually decrease in the case with less mRNA concentration. We can our model can fit well with can predict how the active ribosome fraction change with different ATC concentration. And we can easily extend our model to the growth rate because we showed how mRNA and ribosome determine the active ribosome fraction, and we claimed that peptide elongation rate is constant, so the active ribosome concentration is just the growth rate. So it's very easy to extend our model to growth rate, right? And here we already found our model can fit well with the growth rate in these 4 conditions. And if each cell want to increase growth rate, it have to it's always coordinate mRNA increase with ribosome concentration to set the growth rate. And we have some other tests for our model. First is in this G1 cycling shutoff experiment. After you get rid of all cycling, they're arresting G1, and the ribosome concentration is always constant, like after arrestment, but total mRNA concentration is gradually decreased. So we can predict the growth rates with total mRNA and the ribosome concentration, and our prediction shows that there should be a decrease in growth rate. And we mentioned the local growth rate after arrest. We find it's also decreased. So this can explain why this tells us that the mRNA also influence the growth rate, not only ribosomes. And we have another test because some people may ask, like, if we increase mRNA concentration, how the growth rate changed. Here we do the DCP2 degron system. We add oxime to degron DCP2, which is the decap enzyme of mRNA. So we find after 10 minutes, the total mRNA concentration have an increase in total mRNA concentration and then decrease. And if you mention the growth rate, you can find the growth rate increase in the very beginning and then decrease. Yeah, we can predict the increase of the growth rate, but after that they become sick, so the growth rate decreased. Okay, let's have a simple summary of this content because cell coordinate mRNA. We said cell coordinate mRNA and ribosome concentration to regulate growth rate. Cell has mRNA and ribosome set the active ribosome fraction, and the peptide elongation rates are constant, and so all these set the growth rate. And we have two very important questions of this work because the first is that if we can we increase the growth rate by increasing mRNA concentration at steady state, because the mRNA is very cheap, like it's only 1 or 2% of the dry mass of the cell in a cell. So can we do that? And the second question is like why cell have to coordinate upregulate both ribosome and mRNA to set the growth rate? And for the first question, like we screen a lot of like nature isolates and find that some nature isolates have the very fast growth rate. And look here, the red line is W03 diploid, and these two is are nature isolate. You can find some of them are like 30% or 40% faster than wild type. And if we do spiking mRNA-seq and the mass spec, we find it's just extend this line. It's keep increase mRNA concentration and keep increase ribosome content to increase the growth rate. And so we think we can somehow increase the growth rate by like up regulate mRNA. Yeah. Okay. And at the last, I want to thanks Yang for thanks Yang and my collaborators like Michael Lenz and Rosalind and Puglisi and Kramer for this project. Okay. And do you have any question about this?
All right, fantastic. Thank you very much. As before, if you have any questions, please place them in the chat and we'll come around one by one and get to you. I wanted to start off by asking you if you could just explain again exactly how it is that you're measuring the elongation rates. Because I wasn't quite sure,
Yeah.
maybe just went quickly, whether it's actually something that's being directly measured or is it being inferred from other measurements?
Yeah, it's estimated from other measurement. Yeah, we have the growth rate, we have the active ribosome fraction, and we have ribosome concentration. So it's just estimated from how the protein mass increased in a cell cycle and over the active ribosome concentration. Yeah.
Great. So we have a question from Sandeep. Sandeep, would you like to unmute and ask your question?
Yes. So basically, I was wondering regarding your fits that you did, you showed in one of the earlier slides, where you essentially try to get these two, like, active and non-active ribosome fractions by fitting, a Gaussian mixture model. But for quite a few of them, it looks almost unimodal. I mean, if one looks at the histogram, I mean, yeah, at least, some of them, not all. But so how do you, like, how reliably do you think it then infers these two populations in those cases?
So you mean in some cases it looks like unimodal, right?
Yeah,
So
like
yeah. I think the Gaussian mixture model works quite well. Like if the active ribosome concentration is like between 75% to 25%, but it do have some case like malibars. It's have the like looks like unimodal, but yeah, it's actually not very easy to yep.
I mean, I can imagine that it's really, it's definitely not easy for sure. Yeah,
Yeah, it's very difficult, but we still think there are a fraction of like inactive ribosome. We just use the same method with other conditions. Yeah, but it works well in most of the conditions, right?
Yeah. Thank you.
Yeah.
Thanks.
Great. And so maybe a related question up next from Piyush. Piyush, would you like to unmute and ask your question?
Hi, that was a great talk. I was
Yeah.
wondering if like the inactive ribosomes, when they're bound to like endoplasmic reticulum, like the rough endoplasmic reticulum or outer mitochondrial membrane, have like a
Yeah.
lower diffusion coefficient, and will that like
Yeah.
cause complication in your analysis?
Actually, because if the ribosome is free, it's usually like it's split into 40S and 60 ribosome. We know that some active ribosome, when it's translating, it's will binding to ER, and after it's stopped, translation finished, the 40S will release to the cytoplasm, and the 60S will still attach to ER. But so we we just use the 40s subunit to mention the active ribosome fraction. So it doesn't matter. It's whether the 60s binding to ER or something like that. Yeah.
Okay.
Next up, Bruce.
Yeah, so this is also about measuring the fraction of active ribosomes, and it's similar to the previous question. But two things. In your measurement of diffusion, of the speed of diffusion of the particle, you were assuming, I think, that the RNA is a lot bigger than the ribosome. But that isn't true, I don't think. If the RNA is in a monosome and there's only one ribosome on it, then most of the mass is in the ribosome, right? Isn't that right?
Uh, uh, because we think like ribosome footprint is only like 28 nucleotides, right? The mRNA is on average is 1,500 base pair. And I think that if the ribosome binding with even the monosome binding with the mRNA, it will influence the diffusion coefficient, right? Because
Well,
it's
would
quite
it? Because the ribosome is about 10 times the mass of a 1,000 nucleotide message. So
yeah.
it would only increase the mass by about 10%. But binding of the ribosome to an mRNA would only increase the mass of the whole thing by about 10%.
Yeah, but ribosome you can assume that it's a sphere, but if binding with mRNA, it's no longer a sphere, right? mRNA like it's where something it's so the complex where it becomes a very long mRNA with a ribosome, and we assume that the structure will influence the diffusion coefficient,
Mm.
right?
So also, isn't the amount of free small and large subunits a good reflection of the amount of inactive ribosome, so to speak? I mean, in
Yeah.
other words, what are the monosomes doing if they're not active and on mRNA? Surely when a ribosome gets booted off a message, it dissolves into 40 and 60S subunits.
Sorry, I don't understand. You mean the monosome is like when they don't bind with mRNA, it's what are they doing?
Well, sort of what I'm thinking is that what looks like a monosome, I would have assumed is active. It's
Yeah,
on an mRNA.
yeah,
And when something is inactive, it becomes the 40 and 60S
yeah, yeah,
subunit.
yeah.
But you don't seem to think that.
Uh, uh, we we uh for you mean for the single molecule tracking experiment? Yeah, we consider it because 40S has a faster diffusion coefficient. So you can very easily to distinguish active with inactive, and I think the ribosome because based on model the free ribosome is just waiting for mRNA. It's just like diffused in the cytoplasm and the binding with mRNA. So the inactive ribosome just keep waiting for the collision with mRNA.
Okay, I'm not understanding this very well, but perhaps we should move on.
Okay, thank you.
All right, next up we have a question from Hui.
Hi, thanks for the talk. So my question kind of has two parts. So about your system to manipulate transcription, I guess when you have fewer mRNAs, I imagine ribosomes have fewer binding sites and they would appear more diffuse and thus seem less active. Is there a way you can, yeah, is there a way you can control for that effect when you try to measure the fraction of active ribosomes as a function of decreasing mRNA concentration?
So you mean it will diffuse faster? You think
Yeah,
it will diffuse faster, right?
I
Yeah.
imagine if it has fewer binding sites, then it would naturally diffuse faster, even if you, yeah.
No, actually, I know some paper like study gem, they find the diffusion coefficient may change because it has less ribosome. But actually here gem is very larger than ribosome. And in our system, we didn't find some change of the diffusion coefficient of free ribosome. And because we know maybe the total protein concentration is always constant in there. So even even you decrease the mRNA concentration, the ribosome has the same the maybe same diffusion coefficient. Yeah, we didn't find any difference here.
Yeah. Okay. And about the second part, I think other members of the audience kind of addressed already, but yeah, how likely do you think that there are other changes in the cell that can affect the ribosome diffusion that you see in this system? Like,
Sorry,
yeah, I don't know.
can
Maybe if you can you hear me?
yeah. Say it again.
Yeah. So do you think the change in ribosome diffusion, which we
Yeah,
assume
I
are
understand
the
it.
active function of ribosome, yeah, do you think it
Yeah,
can
I
change
understand
due
it.
to some other effects? Yeah.
Yeah, we didn't actually in our study we didn't find any difference. I understand to you like we just mentioned the diffusion coefficient of active and inactive ribosome in 15 conditions. It's very similar. So if in other case it may change the diffusion coefficient. I know someone did this work, but we it's not the focus of our experiment. Yeah, I have no idea of this. Yeah.
All right. Thank you.
Okay.
I mean, maybe one kind of follow-up question to that is, at least within your model, as I understand it, you're assigning the change in active fractions to the change in mRNA concentrations. But also, for a very long time, people have known that different growth signaling pathways converge to control the activity of translational machinery. So I kind of somehow assumed that must be captured in the way you're thinking about this, but then maybe now I'm thinking back, it's kind of not explicitly stated, like Tor directly controls translational initiation factors, and of course, all of these conditions, Tor signaling would be changing. So is that captured in your kind of the way you're trying to present it?
Tor signaling. Yeah,
all growth signaling, right, which will impact on the activity of translational initiation factors.
Yeah, we actually we haven't think about it. Yeah, we don't know why it's coordinate mRNA with ribosome. We're still confused about it. Yeah, and Tor signaling.
Yeah, well, I guess I meant it in a slightly different way. I mean, it could be the case that Tor is responsible for it in some way, but it could also, it is clearly the case that Tor, signals to inactivate translation initiation factors. So that should reduce the fraction of active ribosomes even without changing mRNA concentrations, if you take all that kind of literature at face value. So that may be also something to
we
consider in what affects these active fractions that you're measuring, right?
Yeah, it's a very good idea. Someone else in our lab, they have studied how Tor signaling influences like active ribosome fraction, but not my project. I'm sorry. Okay.
Okay, thanks. And I think we have another question here from Joel.
Yeah.
Yeah, thank you for your talk. So it's a really technical question. When I'm looking at your the Gaussian fit, I'm actually surprised that it's working so well. Because your mean is so close to 0 and you have so long tails. So like, do you filter out like very low diffusion coefficient values? Like how it is the math tells me that it shouldn't be possible to fit so well. So what do you do? What's the magic here?
You actually know you can see the movie like there's a part of ribosome diffuse very slow. They're almost in the same position, and it's very hard to distinguish extremely slow moving ribosome with inactive ribosome, and we can't ignore. But we have to know that like when it's translating some membrane protein, the ribosome will attach to ER, so this will also lead to the very slow growth condition. So we just use the Gaussian mixture model and do not filter any extremely slow growth condition because we take log of the diffusion coefficient here, and it's very hard to tell the slow growth with ultra slow growth. Okay.
Oh, okay. So you do
Yeah.
not filter, and you do not have values that are around zero. Like
Well, I think we have some ribosome around zero. It's almost zero, but we do not filter it. Yeah.
So, but where are they on your graph then? I mean, it means that your graph goes to zero, not -2, not 0.001.
Oh yeah, it's very slow, but not.
So we're missing. Okay, I think I got.
Yeah.
Answer. Thank you.
It's log 10. Yeah.
Indeed, but we're missing the data that go from 0 to 0.001. That's what I'm saying. So the fit, yes.
But I think maybe very, very small fraction of this.
Yeah, so there's a filter. That's what I wanted to know. Thank you.
Yeah.
Okay, I had another question actually, which is about the DCP2 experiment that you did. And as I was thinking in the Q&A, like degrading DCP2, it stops decapping, but you imagine that especially highly translated transcripts will already be deadenylated. The poly-A tail is kind of at least thought to be important in stimulating efficient translation. So it was a little bit confusing to me how this would then result in the increase in translation that you're proposing that it does. Have you thought about that at all?
Actually, no, because if we degrade DCP2, it has some defect of the MR decay. So we only find the increase of total MR concentration in the first 10 minutes and then decrease. It's where have some feedback to make the mRNA concentration back to the original level. But we can only it's in our experiment only find the growth will increase 10 minutes or 10 minutes to 20 minutes. So it has some feedback, I think.
Yeah, no, I'm aware of that. I think that wasn't really my question. My question was more that DCP2 works at the 5' end of a transcript on decapping, but before transcripts are decapped, they're deadenylated, and that should also inhibit translation. So the transcripts that are accumulating transiently in a DCP2 deletion, it's not clear that they should be translationally competent. So I was just wondering if you'd thought about that and why you think this is working, because intuitively to me, it shouldn't increase the pool of available efficiently translated transcripts for the ribosomes inside the cell. Does that make sense?
Sorry, I don't understand.
So transcripts are normally, typically, not always, but typically deadenylated before they're decapped, right? So deadenylation at the 3' and then decapping at the 5'. So what will accumulate will be deadenylated transcripts, right? Because the transcripts will have already been deadenylated, and then you'll stop decapping, so you'll accumulate transcripts without a poly-A tail in a DCP2 deletion, I think. And so it was just curious to me that within your model that those have some effect, because you wouldn't necessarily expect them to be translationally competent, or maybe they're competent but not translationally efficient.
So maybe this mRNA still have just still not they don't have new ribosome binding to mRNA. Maybe just they have some mRNA just already in there, and if you create new mRNA, it will increase the total mRNA concentration, right? Total active ribosome concentration.
But then it shouldn't affect the growth, right? Or
anyway,
it's something to think about, maybe.
Yeah.
All right. Well, I'd like to thank both the speakers. So that's all the questions that we have today. I'd like to thank both the speakers for two really kind of interesting and thought-provoking talks. Also, a really fun and engaged Q&A for both of them. So thank you, everybody, for taking part. That's the last seminar series for this year, but of course we'll be back next year, and we look forward to seeing you then. Thanks very much. All right. Bye-bye, everyone.